For as long as biology has been taught in classrooms, one idea has been treated as untouchable. Your DNA stays locked inside the nucleus of your own cells, sealed away, private, and completely separate from the DNA sitting inside the cell right next to it. Every cell was thought to run its own genome, minding its own business, never sharing genetic material with its neighbors. That assumption just collapsed. Scientists have now watched, under a live microscope, actual pieces of DNA leaving one human cell and physically crawling into another one, carrying working genetic instructions with them. Nobody expected this. Nobody was even looking for it. And the implications are already shaking up how researchers think about cancer, aging, and the very definition of what makes your cells uniquely yours. Let's set the scene properly, because the discovery came from a place nobody predicted. Researchers at the Children's Medical Center Research Institute at UT Southwestern in Dallas were not trying to prove that DNA can travel between cells. They were studying something more specific, how cells respond when their genome gets damaged or when cell division goes wrong. Errors during division are common, and when a cell mishandles its chromosomes, fragments of DNA can end up floating loose instead of staying properly packaged inside the nucleus where they belong. The team, led by a cancer cell biologist named Peter Lai, was simply watching this process unfold under live cell microscopy, tracking what happens to genetic material after these kinds of division mistakes. What they saw next was not on the research plan at all. Under the microscope, the researchers noticed something strange connecting neighboring cells together, thin, thread-like structures stretching between them like tiny bridges. These structures are called tunneling nanotubes, and scientists have actually known about them for a couple of decades already. They act like transport corridors, allowing cells to shuttle small pieces of cargo back and forth when they briefly come into physical contact. Cancer researchers already knew nanotubes could be used for some fairly dramatic tricks, including cancer cells reaching into healthy neighboring cells and stealing their mitochondria, the tiny power plants that generate energy inside every cell. In some cases, cancer cells have even been observed pushing their own mitochondria into nearby healthy cells, seemingly manipulating them from the inside. So the transport system itself was not new information. What was new, and what genuinely stunned the research team, was discovering that these tubes could carry something far bigger and far more consequential than mitochondria. They could carry actual genomic DNA. When the researchers triggered genome damage in their experimental cells, they watched fragments of DNA physically travel along these nanotube bridges and enter neighboring cells. That alone would have been a notable finding. But the story got even more remarkable once they followed what happened after the DNA arrived at its new address. The transferred DNA did not just sit there as an inert passenger. It made its way into the nucleus of the recipient cell and, in a genuinely startling twist, became incorporated directly into that cell's own genome. The foreign DNA essentially checked in and never left. It persisted through multiple rounds of cell division afterward, meaning it was not a temporary visitor. It became a permanent resident, copied and passed down every time the recipient cell divided, just like the cell's original genetic material. Here is the detail that turns this from interesting into genuinely important. The transferred DNA was not just sitting there as dead weight either. It remained biologically active. The instructions written into that borrowed genetic material could still be read and used by the recipient cell, and in some cases, that borrowed DNA changed how the recipient cell actually behaved, giving it new traits it did not have before the transfer happened. Think about what that means for a moment. A cell's genetic identity, the thing that supposedly makes it uniquely itself, could apparently be altered by material handed over from a completely different cell nearby. That is not supposed to happen according to the textbook version of biology most of us grew up learning. One particular experiment from this study makes the discovery almost impossible to argue with, because it involves a piece of biology that is easy to track with total certainty. The researchers observed fragments of the Y chromosome, the chromosome found in male cells but absent in female cells, moving from male cells into female cells through these nanotube connections. Once inside the female cells, those male-derived genetic instructions were not just present, they were active and functional. A female cell was now carrying and using genetic material that should never have belonged to it in the first place. Because the presence of a Y chromosome fragment in a cell that started out without one is so easy to detect and verify, this experiment served almost like a built-in proof, confirming the transfer was real, not some artifact of contamination or measurement error in the lab. Scientists outside the research team have already started reacting, and their reactions tell you how significant this really is within the field. A cancer biologist at Stanford University described it as an important and exciting discovery, the kind of reaction researchers reserve for findings that genuinely reshape how a field understands itself. And reshaping is exactly the right word here, because this discovery challenges a foundational assumption that has quietly sat underneath decades of genetics research, the the assumption that the genomes inside your individual cells evolve independently of one another. If cells can actively swap genetic material with their neighbors under certain conditions, then that assumption is not entirely accurate. Cells are not necessarily sealed genetic islands. Under the right circumstances, they may be able to directly reshape each other's genomes in ways nobody previously accounted for. Now here is where this discovery gets genuinely important for anyone thinking about their own health, and cancer sits right at the center of the conversation. Cancer, at its core, is a disease driven by harmful mutations accumulating inside a cell's DNA, mutations that let cells divide uncontrollably or dodge the systems that would normally shut down abnormal growth. Historically, cancer researchers pictured this process happening cell by cell, in isolation, with mutations building up independently inside each individual cancer cell over time. But if DNA can physically travel between neighboring cells through these nanotube connections, an unsettling new possibility opens up. Could a cancer cell carrying a harmful mutation actually hand that mutation directly to a neighboring healthy cell, spreading dangerous genetic instructions the same way you might pass a note to the person sitting next to you? Researchers are now asking exactly that question, and it fundamentally changes how we think about tumors developing and spreading. If you're finding this as fascinating as I do, go ahead and smash that like button right now. It genuinely helps more curious people discover this channel. Let's slow down and really absorb what this means for how tumors might grow and evolve over time. If a single cell within a tumor develops a mutation that helps it resist chemotherapy, and that same cell can transfer fragments of its DNA into neighboring tumor cells through tunneling nanotubes, resistance could theoretically spread through the tumor far faster than if each cell had to independently develop the same mutation on its own through random chance. That would help explain something oncologists have puzzled over for years: cases where drug resistance seems to appear across a tumor almost simultaneously, faster than standard models of mutation and cell division would predict. Instead of every resistant cell needing to win its own private genetic lottery, one cell could essentially deal the same winning cards to its neighbors directly. This also forces us to reconsider what damaged DNA really means once it leaves a cell. For a long time, DNA fragments floating outside the nucleus, or worse, outside the cell entirely, were mostly treated as biological debris, byproducts destined to be broken down and cleared away by the body's cleanup systems. This discovery suggests that at least some of that seemingly discarded genetic material is not simply garbage. Under the right conditions, it can find a new home, integrate into a new genome, and keep functioning as if nothing happened. The line between what belongs to one cell and what belongs to another turns out to be far blurrier than biology textbooks ever suggested. It is worth stepping back and connecting this discovery to a bigger pattern researchers have been uncovering about DNA in general over just the last year. Scientists at Northwestern University, working with a large international genome mapping project, recently produced extraordinarily detailed maps showing that the three-dimensional structure of DNA inside the nucleus is constantly shifting, not fixed the way people long assumed. Separately, researchers led by Jesse Dixon showed that DNA repeatedly unfolds and refolds at different speeds throughout the genome, directly influencing which genes switch on and off, with disruptions in this constant motion linked to cancer and developmental conditions. And in an entirely different lab, researchers at the Arc Institute and Gladstone Institutes used a new AI-powered method to discover that the vast majority of the tiny spools your DNA wraps around inside the nucleus are not sealed tightly shut the way scientists believe for decades. Instead, over 85% of these spools show sections of partially exposed DNA, arranged in carefully programmed patterns rather than random accidents. Taken together with a nanotube discovery, a strikingly consistent theme keeps showing up across completely separate research teams. DNA is not the static, locked away, cell-bound molecule generations of scientists were taught to imagine. It moves, it folds, it partially exposes itself in controlled patterns, and now, apparently, it can even leave its own cell entirely and take up residence somewhere else. There is something almost philosophical buried inside this finding too. We tend to think of our genetic identity as something completely fixed and singular, one unbroken code inherited at conception and carried unchanged inside every one of our roughly 30 trillion cells for our entire life. This discovery complicates that picture in a fascinating way. If cells are capable of quietly exchanging fragments of genetic material with their neighbors under stress or after division errors, then the genome inside any single cell in your body, years from now, might carry small pieces borrowed from a completely different cell altogether. Your cellular identity may be less like a single, permanently fixed manuscript, and more like a document that occasionally receives small, unplanned edits contributed by the neighbors sitting right next to it. It is worth pausing on the nanotube structures themselves because they deserve more attention than a passing mention. Tunneling nanotubes are incredibly thin, delicate connections, far too small to see with the naked eye, and they typically only exist for a short window of time before dissolving again. Two cells drift close enough to touch. A temporary tube forms between their outer membranes. Cargo gets shuttled across, and then the connection disappears as if it never existed for years. Scientists studying these structures assumed the cargo moving through them was limited to smaller components, proteins, signaling molecules, and occasionally entire organelles like mitochondria. Nobody seriously entertained the idea that something as large and structurally complex as a chromosome fragment could make that same journey and survive the trip intact. Genomic DNA is not a small, simple molecule. It is long, delicate, and normally protected inside multiple layers of cellular machinery specifically designed to keep it safe. The fact that meaningful chunks of it could travel through a nanotube, arrive undamaged, and still function properly on the other side surprised even researchers who had spent years studying these transport structures. This also raises an interesting question about why nanotubes exist in the first place. Evolution rarely builds elaborate biological machinery without some kind of functional purpose behind it, even if that purpose sometimes gets hijacked or misused. Scientists generally believe tunneling nanotubes evolved as a way for cells to communicate and cooperate, sharing resources during stress, coordinating repair efforts, or supporting weaker neighboring cells during difficult conditions. Cancer cells appear to have found a way to exploit this cooperative system for their own advantage, using nanotubes to drain energy resources from healthy neighboring cells or to spread manipulative signals that help a tumor grow more aggressively. If cancer cells have also learned to exploit this same transport system to spread damaged or mutated DNA, it would represent yet another example of disease repurposing a normal biological tool for harmful ends, turning a system built for cooperation into a system that spreads danger instead. There is also a slower, quieter version of this story worth considering, one connected to aging rather than cancer specifically. As we get older, our cells accumulate more DNA damage and experience more errors during cell division, simply as a natural consequence of years of wear and tear. If DNA transfer through nanotubes becomes more frequent whenever genome damage occurs, then aging tissue, already prone to accumulating more of this kind of damage, might experience more of this cell-to-cell genetic exchange than younger tissue does. Researchers have not confirmed this connection directly yet, but it fits naturally with everything else scientists have been learning about aging on a cellular level. Aging is increasingly understood not as a single steady decline, but as an accumulation of small malfunctions across many separate systems, each one compounding the others. A newly discovered pathway for cells to hand off damaged genetic material to their neighbors would fit right into that broader picture, potentially adding one more mechanism through which cellular problems could spread outward rather than staying contained to the cell where they originated. Consider also what this might mean for how scientists study genetic mosaicism, a term used to describe the fact that not every cell in your body actually carries an identical copy of your genome. Small differences accumulate naturally over your lifetime as cells divide and occasionally introduce copying errors along the way. Researchers have generally assumed these small variations arise purely from internal mistakes made during a cell's own replication process, essentially typos introduced while a cell copies its own DNA. This discovery introduces a completely different source of variation to consider. If cells can directly receive foreign genetic fragments from their neighbors, rather than only generating internal copying errors, then some of the genetic differences observed between cells in the same tissue might not be internally generated typos at all. They might be borrowed material, handed over from a nearby cell during a moment of stress or division error, then permanently absorbed into a new genetic address. Untangling which small genetic variations come from internal mistakes and which ones come from this newly discovered transfer process is going to keep researchers busy for a long time. There is a reason discoveries like this tend to ripple outward far beyond the specific lab that made them. Once one team demonstrates that an assumed biological boundary is not as fixed as everyone believed, other researchers across completely different specialties start re-examining their own data with fresh eyes, wondering whether this newly discovered mechanism might explain patterns they had previously struggled to account for. Immunologists, studying how the body distinguishes its own cells from foreign invaders, may need to reconsider what happens when a cell is quietly carrying borrowed genetic material from a neighbor. Developmental biologists, studying how tissues form and organize themselves, may start asking whether this kind of cell-to-cell genetic exchange plays any normal role during early development, separate from disease or damage. Even researchers working on aging and regenerative medicine may find themselves revisiting old assumptions, wondering whether encouraging or blocking this newly discovered transfer pathway could eventually become a tool for treating disease rather than just a phenomenon to study from a distance. There's also a reasonable question worth asking here, which is whether this kind of transfer could ever work in a beneficial direction rather than only a harmful one. Most of the framing so far has understandably focused on cancer and disease, since that is the context in which the discovery was made. But biological mechanisms rarely serve only one purpose. If damaged or mutated DNA can move between cells and get incorporated into a new genome, it stands to reason that healthy, functional genetic material might occasionally make the same journey under the right circumstances. Some researchers are already speculating about whether this pathway could eventually be harnessed deliberately, engineered to deliver corrective genetics, material directly into cells that need it, essentially borrowing a trick nature already figured out on its own. That kind of application is still speculative and years away from any real testing, but it illustrates how quickly a discovery like this can open up entirely new lines of thinking, moving beyond simply explaining a problem toward potentially engineering a solution. It also helps to remember how discoveries like this one tend to unfold over time. The initial finding rarely arrives as a complete, tidy explanation. It arrives as a genuinely surprising observation, confirmed carefully enough to be trusted, and then followed by years of additional research, chasing down every branching question it raises. The scientists behind this particular study were not chasing this outcome when they began. They were simply documenting how cells respond to genome damage, using careful microscopy to track a process most researchers assumed they already understood reasonably well. What they stumbled onto instead was a completely unexpected transport system for genetic material, hiding in plain sight the entire time, visible only because someone happened to be watching closely enough, at exactly the right moment, to notice something moving that was never supposed to move at all. That is often how the biggest discoveries actually happen in real science, not through some grand plan to overturn an established idea, but through careful, patient observation that happens to catch nature doing something nobody predicted. The tunneling nanotubes had been sitting there the entire time, known to science for roughly 2 decades, quietly connecting cells and shuttling smaller cargo back and forth without anyone realizing they were also capable of carrying something as significant as functional chromosome fragments. It took a specific set of experimental conditions, a dedicated research team, and a fair amount of scientific luck for anyone to actually witness DNA making that journey and catch it clearly enough to prove it beyond doubt. Of course, there is still an enormous amount left to figure out, and the researchers themselves are careful about that. This transfer process was triggered under specific experimental conditions, involving genome damage and cell division errors, and scientists still need to determine exactly how often it happens naturally inside a living human body, under normal day-to-day conditions, without any artificial triggering in a lab dish. They need to understand which cell types are most prone to sending or receiving this kind of genetic cargo, whether the immune system normally polices and eliminates recipient cells that pick up foreign DNA, and whether this mechanism plays a meaningful role in healthy tissue, or mainly shows up in disease states like cancer. These are exactly the kinds of follow-up questions a genuinely important discovery is supposed to generate. More open doors than closed ones. But even with all of those open questions still sitting on the table, the core finding itself is already remarkable enough to change a fundamental assumption in biology. Human DNA does not have to stay locked inside the cell where it started. Under the right conditions, it can escape, travel along a temporary bridge built between 2 neighboring cells, move into a brand new nucleus, and quietly become a permanent part of a completely different genome, continuing to function and shape that new cell's behavior for the rest of its existence. That single sentence would have sounded like science fiction to most cell biologists just a couple of years ago. Now it is a peer-reviewed result published in one of the most respected journals in the field, watched happening in real time under a microscope, impossible to dismiss as a fluke. Before you go, if discoveries like this genuinely fascinate you, subscribe to the channel so you never miss the next major breakthrough the moment researchers publish it. Biology keeps proving again and again that the systems we assumed were fully understood are still hiding entirely new layers of behavior beneath the surface. Your cells may be talking to each other, trading genetic material, and quietly reshaping one another in ways no one even suspected until a handful of researchers happened to notice something strange stretched between 2 cells under a microscope. The genome inside you right now may not be quite as fixed, quite as private, or quite as finished as you were always led to believe.